Good morning, everyone, and welcome to the 8th Single-Cell Proteomics Conference. It's a great delight to see you here and open the meeting for this year. As we all know, what brings us together is our interest in understanding biology, not just at the level of the genome and the transcriptome, but also embracing the complexity at the level of the proteome with the myriads of proteoforms, modifications, dynamics in time and space. And of course, paying very close attention to all of the other relevant layers of biology, including the regulation of transcription, RNA metabolism, modifications, and all of these other processes that ultimately impinge on and shape the proteome. In this meeting, we are focused on solving biological questions, and we are agnostic to the methodologies that we use. Of course, I'm very enthusiastic about the capabilities of mass spectrometry proteomics, but I'm equally appreciative and very enthusiastic of the awesome capabilities of imaging that until this day has allowed us the highest time resolution of being able to see protein dynamics. And as Galit Lahav is going to tell us, that's really, really important to understanding much of what makes us alive. And as customary, we are going to have presentations in this meeting from colleagues working in the realm of single molecule proteomics, emerging technologies. We are going to have presentations from mass spectrometry proteomics, and of course, from methods using affinity reagents, fluorescent proteins, and so on. And ultimately, I think that there is a lot that we can learn from those different domains, from these different technologies. As we made the argument in this perspective a couple of years ago, together with Mike McCoss, many of the challenges that those different technologies face and try to resolve are similar, such as the immense scale of the proteome. And approaches that have been helpful in one domain might also help in another domain. So in this particular case, we suggested that using separation that has long been the mainstay of mass spectrometry analysis can also be very beneficial for single molecule proteomics. And I think that there are many such examples. So one thing that I'm very proud of this meeting is that it brings together people who rarely attend the same meeting. And I think that's a catalytic capability there for us to all try and understand the developments in another area. We started in 2018 on this campus, and I think it's worth taking just a minute to recognize how much the landscape has changed. What precipitated the first meeting was our excitement around the capabilities to start making more specific, more scalable measurements with mass spectrometry. And at that point, there were some enthusiasts like us and Ryan Kelly who thought that this was indeed feasible, and we were excited to start developing it. And there was a lot of skepticism that maybe all that we are measuring is noise, and proteins are still beyond the sensitivity of mass spectrometry. And by now, all that skepticism has dissipated. So in 2018, part of what we were trying to do is to demonstrate the quality of our measurements in vitro grown cell line cells. And today we are going to have presentations from colleagues analyzing many thousands of primary cells from complex mammalian tissues and measuring not just protein abundances, but rates of synthesis and degradation modifications, and not only measuring things, but really interpreting modes of regulation. And I think that's amazing progress for somebody who has witnessed that, who has seen this meeting changing from 2018 to now. It's remarkable. It has certainly taken a lot of effort from our community, from all of you and many, many people beyond this room. But for me as a participant of this movement, it's really amazing and encouraging what we have been able to do and how much more we have ahead of us, because the rate of progress, if anything, is accelerating. So as we took picture in 2018, we're going to take another picture today, and that is going to be just before lunch. As I already mentioned, we have colleagues joining us with very different backgrounds. Some people are very focused on biomedical research, biology, medicine. Other people are the foremost experts in their technology realm. Other people are computational biologists or mathematicians. And our very lofty aspiration, and I'll confess, it's very, very hard, but our aspiration is to make the presentations as accessible as we can possibly make them to everyone. Try to explain things conceptually, and ultimately, we are going to need assistance from the audience. You need to ask questions. That will be relatively easy because you'll see that everybody does that. That's usually the ethos of this meeting. We like to keep it very interactive, very engaging. And this is just, for those of you who are first-time comers, this is just the first time that you're going to hear it, and soon you're not going to need to hear it because it will be obvious. But the goal is really to make everything accessible and to discuss. It is not so much to impress with all of the amazing results that we have. I know that there will be a lot of amazing results, but what I think is very special about being here together is that we can have discussions. We can read our papers. Many people can watch us online, which is wonderful. We have a large audience to which we are streaming the meeting, and I'm very happy to make it accessible. But what I think is very special for us being here is that we can interact with each other and ask very spontaneous questions. And that's why we have plenty of time after presentations, during breaks, in between breaks, to talk, and lots of good food. I'll get to that in a minute. So the goal is to be able to create a community that is very interdisciplinary and that as a whole possesses all of the skills and expertise that we need to advance this field forward. None of us does, but together we can help each other to make progress in what is admittedly very challenging, very daunting area of research. There's just no getting around that proteins and their interactions and modifications are very, very complex, and we still understand very little of that, which is exciting. I already emphasized how much we love questions. For those of you who are joining us virtually, I would ask you, if you're not asking questions or presenting, to turn off your video and audio so that there are no interruptions. We're going to have many excellent meals throughout the conference, including dinner for all attendees tomorrow evening. That's at 5 Horses, a short distance walk from campus. We can all go together. And that will be a very nice informal location where we can mingle and interact with each other. It's going to be a buffet-style dinner, and I think we're going to have a lot of fun with it. We also will have a number of posters presented today and tomorrow, and we are going to have poster prizes. I'm not going to tell you who are the judges, so that you try to impress everyone, not only the judges, but I'll tell you that the prizes are cool. All right. And of course, it's very, very important to acknowledge the support of our sponsors who have enabled all of the lovely meals that we're going to have throughout this conference. Thank you for supporting us. And I'm looking forward to a very exciting meeting with lots of biology and lots of technology and lots of interactions between those different domains. So I'd like to invite Keller to introduce our first speaker in our morning session, Professor Nikolai Slavov, from not far away, just across the road from Harvard Medical School. Keller, please.